Welcome dear students. Today we shall talk about the origin, evolution and processing of jute. Corchorus is a genus of annual herbs belonging to the Tiliaceae family. Only two species, Corchorus olitorius and Corchorus capsularis, are well known for commercially bass fiber production. Jute is a kind of fiber which is obtained from two species, Corchorus capsularis and Corchorus olitorius, of the genus Corchorus belonging to the family Tiliaceae. It is one of the cheapest and most economical vegetable fiber after cotton obtained from the skin or bast of the plant stem. It is recyclable, 100% biodegradable and eco-friendly. Jute has low extensibility and high tensile strength. Jute is the versatile natural fiber widely used as a raw material in many textile, non-textile, packaging, construction and agricultural applications. It has been used since ancient times in Africa and Asia to provide a cordage and weaving fiber from the stem and food from the leaves. Several historical documents such as Ain-e-Akbari by Abul Fazl in 1590 during the era of Mughal Emperor Akbar states that the poor villagers of India used to wear cloths made of jute. Quality of jute fiber has direct relationship with the stage of harvest. The plants usually harvested at the early flowering stage in order to obtain good quality fiber are really in great demand. There are two stages of grading jute fiber, one for the home trade and another for the export trade. Grading factors for jute are color, length, firmness of fiber, luster, strength, clearness, free from defects, and the amount of root end which will have to be cut off. The average yield of jute fiber is 2.07 tons per hectare. However, with improved package of practices, it is possible to get 2.53 tons per hectare of fiber. On the basis of the two criteria, that is amount produced and types of uses, jute is considered as the second most important fiber producing plant throughout the world and as well as in the Indian subcontinent after cotton. The jute fiber is used mostly for making gunny bags and packaging material for agricultural and other industrial products. Jute plays a vital socio-economic role in producing countries. About 12 million farming families, mainly in Southeast Asia, are dependent on this crop. Out of our 30 important species belonging to the genus Corchorus, only two, that is, Corchorus capsularis, commonly known as white jute, and Corchorus olitorius, known as Tosa jute. Now we shall talk about common names and synonyms of jute. Jute was previously named put, and the first commercial export of raw jute to England was under this name. The first commercial application of the word jute appeared in the custom returns of the 1828 exports, leading to suppression of the word put ever since. Now let us talk about species diversity. The Index Caviensis includes more than 170 species of Corchorus and based on species concentration. East and South Africa were considered to be the center of diversity and place of origin, though both the cultivated species of jute are distributed throughout India. The genus Corchorus is extremely variable, but all species are highly fibrous. 10 species, 2 cultivated and 8 wild, are reported from India. The species include Corchorus capsularis and Corchorus olitorius, Corchorus aestuans, Corchorus depressus, Corchorus fascicularis, Corchorus pseudo-olitorius, Corchorus tridens, Corchorus tricularis, Corchorus urticifolius and Corchorus volutinus. Corchorus capsularis or white jute and Corchorus olitorius, that is Tosa jute, yields fiber of commerce from bark of the stem, that is phloem fiber. Jute is distributed throughout the world's tropics, subtropics and warm temperate regions. Jute is widely cultivated in India, Bangladesh, Nepal, China, Indonesia, Thailand, Myanmar and in South American countries. The major jute growing countries are India, Bangladesh, China, Thailand and Nepal. The fiber obtained from the cultivated species is retted in water and termed as jute. Although the wild species of jute are poor fiber yielder, but they are genetic resources for abiotic stress tolerance, disease resistance and fine fiber quality. Germplasms of wild species of jute are exploited for efficient breeding endeavor with the cultivated members for crop improvement. The germplasms are mostly evaluated for potential donor for desirable traits, although utilization of trait-specific germplasms have increased the productivity, but with the advent of synthetic material, jute faces serious competition in the market. Now let us talk about distribution of jute. Jute is grown in Bangladesh, India, Myanmar, Nepal, China, Taiwan, Thailand, Vietnam, Cambodia, Brazil and some other countries. Quarkorous germplasm occur in diverse ecological conditions and habitats like riverbanks, dry riverbed, low altitude valley within mountain folds, hill cliffs, forest floors with open canopy, marshy land, roadside fallow, ditches, cultivated as well as homestead lands. The major jute growing states in India are West Bengal, Bihar, Assam, Uttar Pradesh, Meghalaya and Tripura. Quarkorous capsularis is frequently distributed in northern parts of India and gradually becomes scarce towards west. In contrast, Quarkorous oletorius is more frequent in western and northwestern India. Quarkorous tridents and Quarkorous tricularis are restricted to central, western and southern part of the country, whereas Quarkorous fascicularis is restricted in western and peninsular India. Quarkorous urticifolius is restricted in Tamil Nadu of southern India, while Quarkorous pseudo-oletorius is distributed in western boundary of the country. Quarkorous capsularis is considered to be native to South China, from where it migrated to India and Bangladesh. However, some believe that this species is not an emigrant to India but had originated in Indo-Myanmar region, including South China. In contrast, Quarkorous oletorius is proposed to be native to Sri Lanka, India and Kenya, and it is now agreed that the species originated in Africa along with other wild species and migrated to India and China via Egypt and Syria. The diversity and worldwide distribution of jute has now been well recognized as economic resource, providing ample scope to mankind to identify and utilize plants, especially for food, fiber and other needs. Now let us talk about cultivation of jute. It can be grown in any intermediate soil other than the extreme sand and extreme clay soil. The jute growing areas of the world are found spread over several degrees north and south of Tropic of Cancer and generally between longitudes of 86 and 92 degree east. The crop responds favorably to high relative humidity of 70 to 74% and annual rainfall of 1500 millimeters or more, with at least 250 millimeters of monthly precipitation during each of the months of March, April and May. The requirement in regard to range of mean temperature is 18 to 33 degrees centigrade, having sandy loamy soil, organic carbon 0.76%, soil pH 6.85 with elevation level of 48 feet above mean sea level. Land preparation is very important for successful cultivation. Cross plowing and laddering is necessary to prepare uniform smooth soil to have 20% organic content. Generally, seed bed is prepared by repeated drilling, plowing and leveling to produce finely macerated and well aerated and cold free soil. Manuring may be done during plowing. Mostly cow dung is used along with nitrogen, phosphorus and potassium in appropriate proportion according to the soil type. However, when fertilizer is used, it must be applied in 3 stages. 1. At land preparation and 2. As top dressing at appropriate time. During cultivation, weeding is usually done in addition to thinning. Now, let us talk about plantation of jute. Generally, the species is propagated by seeds either by broadcasting method, that is 10 to 12 kg per hectare, or by line sowing, lower amount of seed is required. Keeping distance between plants, that is 5 to 10 centimeter, and rows 20 to 25 centimeter. Lack of proper plantation strategy leads to poor utilization of field and thus responsible for reduced yield of jute. It is therefore very much necessary to choose a right option for plantation among others. Now, let us talk about germination of seeds. Seed dormancy creates a major difficulty in Quercus cultivation. There is faster rate of imbibitions of Quercus capsularis seeds than Quercus oletorius under different degrees of water stress, resulting in the earlier germination of Quercus capsularis seeds. Under field condition, seed germination and establishment are most obviously affected by soil water deficit, resulting in poor crop stand. There is wilting and early flowering tendency of young jute seedlings at low humidity below 19% with soil moisture less than 20%. The optimum temperature for seed is 25 to 30 degrees centigrade. Treatment of Quercus seeds with concentrated sulfuric acid, that is 98%, for 10, 20 and 30 minutes significantly increased the germination compared to other treatments. The hot water treatment have a positive role in seed germination but with lesser efficiency. Seed germination is one of the crucial aspects in plant breeding. Most of the time, plant breeders as well as researchers faces failure in germinating seeds due to various constraints like deep seeding, planting in cold soil, extremes of watering, improper soil preparation, birds or squirrels, or insect activity and poor seed quality. Therefore, a proper strategy or combination of more is needed to overcome the problem. Now, let us talk about the yield of jute. The use of old jute seeds with poor germination capacity gave poor stand and consequently low yield. But the yield of individual plant which constitute the crop in an average are comparable to those raised from fresh seeds. The water stagnation in the field has been found to possess a lasting effect on jute yield and the quality. The impaired seedling establishment was found to be the single most important edific factor for the year-to-year fluctuation in jute yield. The seedling vigor as well as plant growth and subsequent yield have a positive correlation with seed size. Treating seeds with garlic extract 1:2 ratio, weight by volume per gram per ml, and Vitavax 200 increases the yield and reducing several fungal attacks. Better root growth in Corchorus capsularis varieties than Corchorus oletorius under typical rain-fed and moisture-stressed upland situation in early stages of growth. The seed yield of Corchorus is influenced by sowing time, seed rate, and sowing method. Corchorus capsularis have been shown to produce an initial higher rate of dry matter production than those of Corchorus oletorius with the advancement of growth. The plant height and diameter have a direct relationship with the yield of Corchorus oletorius and Corchorus capsularis. The final goal of any kind of cultivation, including jute, is getting better yield performance in the field. Low yield is associated with poor germination of seed, impaired seedling establishment, improper sowing time and method, poor photosynthetic activity. Now let us talk about retting process. Retting is a biological process by which the bast fibers are extracted by decomposing the plant by the combined action of water and aquatic microorganisms, mostly bacteria. The process of separation and extraction of fibers from number of fibrous tissues and woody part of stem through dissolution and decompositions of pectins, gums and other mucilaginous substances is called retting. In general, retting is the mechanical extraction, washing, drying, followed by marketing of fibers. Retting is a kind of fermentation process in which the cortical and phloem tissues of the bark of the plants containing the fiber strands are decomposed by the joint action of microorganisms, mostly bacteria and water. Immediately after steeping the plants under water, the soluble carbohydrates, proteins and minerals start leaching out of the plants and water becomes very rich in aerobic bacterial contents. The water together with these bacteria enters the plant tissues through stomata, replacing the intracellular air. This water together with these bacteria enters the plant tissues through stomata, replacing the intercellular air. At this stage, because of the fermentation of the soluble carbohydrates, organic acids are formed, pH decreases and the retting water becomes very low in oxygen content. Butyric acid, acetic acid, lactic acid have been identified as some of these acids. In such a condition of retting, mostly microaerophilic bacteria start multiplying, entering the tissues and decomposing the cementing materials, mostly pectineous in nature, connecting the fiber strands. At this stage, the pH value increases because of the formation of buffering organic compounds and the retting of the plant is completed. The enzymes by which the microbes actually carry out the decomposition are pectin polygalacturonase and pectin methyl esterase. Now, let us talk about conventional methods of retting. Conventionally, for the extraction of fiber, the plants are retted in the water of lakes, rivers, ponds, ditches and roadside canals. Harvested plants are made into bundles weighing about 10 pounds each, steeped under water in layers. Stems of banana plants, mango logs, earth chunks and water hyacinth are used as weights for submerging the bundles of the plants known as jacks under water. It takes about 15 to 20 days to complete the retting. The fibers are then manually extracted, washed and dried either by beat, break, jerk or single plant extraction method. Now let us talk about microbial retting. It appears that most of the retting water contain aerobic, anaerobic and microaerophilic bacteria, aquatic and other fungi, algae, protozoa and diatoms. Of all the microorganisms, aerobic and anaerobic or microaerophilic bacteria have been found to be mostly involved in jute retting. The most predominant ones of such bacteria have been found to be Bacillus polymixa, Bacillus subtilis, Bacillus macerans, Bacillus pumilus, Bacillus sphaericus, Clostridium butyricum, Clostridium tertium, Clostridium lacunarum, Pseudomonas putida, Pseudomonas aeruginosa, Pseudomonas pseudomolii, Micrococcus casiolyticum, Micrococcus corcorus, Micrococcus lutens, Micrococcus varians. These bacteria completed retting in sterile water under laboratory conditions in 2 to 10 days, whereas normal retting time under growers condition is 15 to 20 days. In case of fungi, mostly the non-aquatic ones as Aspergillus niger, Penicillium fructans, Mucor species, Sclerotium rolfsi, Mycelia sterilia have been found to ret jute under moist condition and spatial environments of temperature and nutrients in about 3 to 10 days. Algae, protozoa, diatoms, which are usually present in the surface layer of water, perhaps indirectly participate in the retting done by microaerophilic bacteria by providing appropriate environments, since retting progressed faster in surface layer of water. Attempts have been made to make use of the active retting bacteria and fungi at growers level, but their activities were not quite as encouraging as in the laboratory, since perhaps in non-sterile water at growers conditions, the presence of other bacteria inhibited their sufficient multiplication. It seemed possible to undertake large-scale retting with the isolated active retting fungi, but this again calls for industrial approach under controlled conditions. Furthermore, fungal retted fibers are rather coarser than normal retted fibers. For the acceleration of retting speed, other methods are also used, such as number 1, retting effluent, number 2, suitable economic chemical agents serving as nutrients for the retting microbes, and number 3, other plant materials. Retting effluents are usually very rich in retting bacteria. It was found that if these effluents were added as inoculum to the retting water at the time of steeping of the plants at the rate of 1% of the green plant, retting speed was accelerated. Among the number of chemicals, that is urea, ammonium phosphate, ammonium oxalate and ammonium chloride were used. Urea greatly reduced the retting time by maintaining the pH value at about neutral level and increasing the bacterial population greatly. Retting could also be completed in hours using ammonium oxalate solution at about 90 degrees centigrade, but this method is considered costly. The addition of water hyacinth and pistia in retting reduces the retting period by about 5 days and improved the fiber quality. Besides, the addition of blue-green algae also enhanced the retting speed by several days and improved the fiber color. Now let us talk about the diseases and pests of jute. Jute crop is attacked by various diseases and insects under field conditions. Stem rot, anthracnose, wilting, soft rot, dieback, black band and powdery mildew are the common fungal diseases of jute. Hairy caterpillar, semilooper, stem weevil, yellow mite, and the indigo caterpillar are the major pests of jute. Jute productivity suffers vastly due to the ravages caused by many insect pests, and hence the entomology of bast fiber crops requires much attention for economic production. The species A. subulifera is a major pest of crop which occurs in all the jute growing sectors of India. Repeated damage by this pest checks crop growth and induces profuse branching with reduction of fiber yield. A third instar larva of A. subulifera is more penetrative in action, and the larval period ranges between 9 and 16 days. Another most important pest of the jute is stem weevil, Apium corcori, that exists throughout the cropping season and damages the early sown crops. Corcorus capsularis is more susceptible to stem weevil infestation, whereas Corcorus olitorius suffers little because of its higher tannin content. Yellow mite causes serious damages of jute since the 1940s. Walker, Spilosoma obliqua, once considered as a sporadic pest on jute, is now a major threat to jute crop. Other pests of jute like semiloper, stem weevil, Apion corcori, martial, yellow mite, Polyphago tarsonemus latus, indigo caterpillar, Spodoptera exiguus, Bihar hairy caterpillar, Spilosoma obliqua. A number of minor pests, scale insects, Pinesapis, thrips, Aeria, mealybugs, Fericea of jute are also reported. Grey weevil was the first time recorded as a pest of Tosa jute in 1973, and the crop loss due to the pest was estimated at up to 50%. The cultivation of jute is severely affected due to disease and pest attack. This plant type is a host of different pests and disease causing microorganisms, and the nature of damage is due to degree and intensity of infections. If the breeder is not familiar with the pest, then the nature of damage will also be unknown, and no preventive measures can be adopted. For this purpose, detailed knowledge on different jute pests and disease regarding the cause of infections, nature of infection, life cycle, and previous reports of the damage is necessary before formulating any preventive measure. Now let us talk about integrated pest management. JRO 524, also known as Naveen, and JRC 212, also known as Vasudev, are recommended for cultivation in the yellow mite endemic areas as the least susceptible varieties. Studies on seasonal incidence and population dynamics of major pests of jute revealed that the intensity of damage by stem weevil, grey weevil, and yellow mite were more on early sown crops, but the reverse was true for semi-looper incident. Both Tosha and white jute varieties sown in the late April show less incidence of stem weevil as compared to those sown in late March to early April. Physalis minima was reported as an alternate host for yellow mite. There is a negative impact on the gut physiology of semi-looper larvae due to ingestion of Bacillus thuringiensis. Some insecticides like neem oil, endosulfone, cypermethrin are effective against jute stem weevil and semi-looper. The incidence of stem weevil was observed to be less in jute plants inoculated with rice necrosis mosaic virus at early stage of growth of 20 days after germination, irrespective of the fertilizer regimes on both Tosha and white jute varieties. Although few parasitoids were reported as effective biocontrol agents on jute pests, but the mass culture technology is yet to be perfected, and thus insecticidal interference is unavoidable in spite of their negative impact on the ecosystem. Besides the application of insecticides, regular pest surveillance is essential. Advanced integrated pest management strategies on the basis of previous damage reports and responses after application of pesticides and other measures are required for achieving further success. Finally, to wind up the lecture, it is important that the growers are motivated to produce quality jute fiber. To save jute going the indigo way, there is a requirement for development of high yielding varieties, development of more location specific retting technologies, production of cutting free good quality fibers, and assurance of fair price for the jute fiber. So dear students, it was all about today's lecture regarding origin, evolution and processing of jute. Hope you understood it well. So see you next time with a new topic. Till then, take care and goodbye.